A cell in interphase, having received a chemical signal telling it it's time to divide, moves from G1 into S. There, the cell continues to grow, synthesizes new DNA and new histone proteins around which the DNA is wrapped to form new chromatin. The cell then moves to G2, where it continues to grow and it makes final preparations for cell division, replicating numerous organelles and making sure that the DNA is in proper order and is free of errors. The cell then enters the mitotic phase where active division will occur.
Cell reproduction is cell division. It's how cells reproduce. We start with one parent cell, and it goes through a process that results in the production of two what we call daughter cells, which are identical to the original parent cell. Why is cellular reproduction important? Well, it's absolutely essential for growth and development. One of the mechanisms by which humans grow is by the production of new cells. Cellular reproduction is also required for the repair of damaged tissue. When you are injured, cells are destroyed and they die by a process called necrosis. And you don't have to know that for lab, but we'll touch on it in lecture. The healing process from an injury involves replacing the cells that were destroyed with new cells. So cellular reproduction or cell division is required for repair of damaged tissue. And finally, there are certain cells in the body that just don't have very long lifespans. Your skin cells, for example. Your skin is constantly being sloughed off. In fact, the majority of the dust in your house is really dead skin cells. Red blood cells only live about 120 days, so they have to be replaced. And white blood cells, especially if you're fighting an infection, can literally live for just a few hours. So it is necessary in order to replace cells that just naturally die for cellular reproduction to occur. Natural cell death, by the way, is apoptosis, as opposed to necrosis, which is abnormal cellular death. So cellular division or cellular reproduction is absolutely essential for growth and development, repair of damaged tissue, and replacement of cells that die naturally. Let's take a closer look at cell division or cell reproduction. We start with a parent or mother cell. We end with two new daughter cells which are identical to the original parent cell. Identical in every way, down to every DNA molecule. Let's think about what has to occur. What are all the events that have to occur in order for us to get from this one parent cell to two daughter cells that are identical to the original parent cell? Well, obviously, and I like to start with the end and work backwards. Obviously, the cytoplasm is going to have to split apart. And you can see it beginning here with the development of what's called cleavage furrow. Then we have a deeper cleavage furrow. And then eventually the cytoplasm breaks completely when you have the two new daughter cells. That is cell division. And that process is called cytokinesis. Kinesis refers to movement. Cyto refers to cell. So cytokinesis is literally the splitting apart of the cytoplasm. That is what cell division is, cytokinesis. Cytokinesis is cell division. Often, students are taught that mitosis is cell division. It is not. Cytokinesis is cell division. Where does the mitosis come in? Well, let's think about what has to occur before the cytoplasm splits apart. If we're going to go from one parent cell to two daughter cells that are identical to the original parent cell, don't we have to duplicate everything? Don't we have to duplicate the mitochondria and the rough endoplasmic reticulum and the ribosomes, etc., etc.? Yes. Don't we have to duplicate all of the DNA? Yes. In fact, that process is called replication. So when DNA is duplicated, it's replicated. That is mitosis. So after there is replication of the DNA, mitosis makes sure that exactly one copy of every DNA molecule gets into the two new daughter cells. We don't want two copies of one DNA molecule to get into one daughter cell and none into the other, right? We want exactly one of each copy to get into one of each of the two new daughter cells. And that is what mitosis accomplishes. That is what's occurring all along here. After that DNA is replicated, so there's two copies of each one, mitosis makes sure that exactly one copy of each chromosome is made part of the two new nuclei of the two new daughter cells. So mitosis is not cell division. It is a related process that is required for successful cell division or cytokinesis. Mitosis is division of the nucleus and segregation of the DNA. Cytokinesis is actually cell division. In this lab, you will be identifying the different stages of a cell's life cycle. You'll be identifying cells in interphase and the different stages of mitosis: prophase, metaphase, anaphase, and telophase. You'll also be identifying cytokinesis as well. You'll be identifying these stages on a microscopic slide, an image, or on various models. I have found that if you're new to cell division, it is helpful to at least be able to put the different stages of a cell's life cycle in order. And to do that, it might be helpful to use a little mnemonic device. And one that I came up with that I thought every student could relate to was IP at MATC. If you are on campus, well, before we went online, if you are on campus long enough, you're probably going to be drinking beverages, and from one time or another, you have to stop by the little boys' and little girls' room and pee. So I thought IP at MATC could be something everyone could relate to. And what does that mnemonic mean? Well, it stands for interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. It's just a way to at least order those different stages in your mind. Just remember, however, that mitosis only includes prophase, metaphase, anaphase, and telophase, and that cytokinesis is a separate process, the process where the cytoplasm splits apart, cell division. Let's take a moment to talk about your learning objectives. So what are you expected to be able to demonstrate as part of this lab? You're looking at a mitosis model on the left. You're looking at a slide of a cell undergoing mitosis on the right. You are expected to be able to correctly identify the stage of mitosis or other portion of the life cycle of a cell, such as interphase. Both the model and the slide are showing anaphase. You're also responsible for being able to identify any other structure that's listed on your study guide that is visible on a model. such as chromosomes. You should also recognize that those dark stained structures are chromosomes on a slide as well. But all the other structures, I don't typically ask for those to be identified on a microscopic slide, but certainly you should be able to identify them if they're visible on a model, such as mitotic spindle, centrioles, etc. Let's talk now about study skills as they apply to learning mitosis. What I find is the default method of study for students is to memorize. Memorize, memorize, memorize. I would recommend against memorizing in order to learn successfully to identify the stages of mitosis. Instead, recognize that mitosis is a process. Study and learn it like a process. If you do that, it'll make more sense to you, you'll learn it more quickly, and you will be very successful. Let me explain what I mean. So this is a figure I've modified from your textbook. Let's look here at this cell. This cell is at interphase, so it hasn't started mitosis yet. How do I know that? One, I have an intact nucleus. I will never have an intact nucleus during mitosis. Two, I have two centrioles sitting right beside one another, like they're hanging out. Okay? Again, I will not have centrioles sitting beside one another during mitosis. So I know that we are in interphase. What is the very first thing that happens during mitosis? Well, the very first thing that happens is the centrioles start moving away from one another. They start moving to opposite ends, and the nuclear membrane, or nuclear envelope rather, begins to break apart. So I know I'm at the very first stages of mitosis, which is prophase. Eventually, that nuclear membrane or envelope breaks apart completely, and it releases the chromosomes. This is also still prophase. Okay? I like to think of this process of the nuclear membrane breaking down and releasing the chromosomes, since we're in Wisconsin, like going to a dairy farm and knocking down the gates and releasing the cows, and the cows go, moo, we're free, and they start roaming all over the countryside. Well, the chromosomes start roaming all over the cytoplasm. But as any good Scotty knows, the cows always come home, and the chromosomes do as well. Those chromosomes come home to the equator of the cell, the central portion of the cell, also referred to as the metaphase plate. And at that point, we are in metaphase. Metaphase is the easiest stage of mitosis to identify. If you miss metaphase on a quiz or on an exam, 50 lashes with a wet noodle. So what happens after those chromosomes line up at the equator? The centrioles, using the spindle fibers, pull tension and literally pull them apart. And when they are pulled apart, we are now at anaphase. Remember, ana means apart, like anatomy, tomy, to cut, ana, apart. Ana means apart. And as long as those chromosomes are still moving to the opposite ends, that cell is in the stage of anaphase. Eventually, those chromosomes reach their destination, and the new nuclear envelopes begin to form, and at that part, we are at the end stage, which is telophase. Also note that we have a second process that has started, which is cytokinesis. We have a cleavage furrow. That's the structure. The process is cytokinesis. Let's use the same process as we look now at the models. Here we have a cell at interphase. I know it's an interphase because I have an intact nucleus and the two centrioles are right beside one another. Here I have a cell at prophase. How do I know? Our centrioles have moved toward the opposite poles, and my nuclear envelope's beginning to break down. Eventually, the nuclear membrane or envelope has completely broken down, and my chromosomes have been released. And so we are still at prophase. Students often ask the question, do we have to distinguish between early and late prophase? And the answer is no. You only have to identify the stage as interphase, prophase, metaphase, anaphase, and telophase. Some phases are longer than others, and so in explaining the process to you, they'll split them up into early and late, but you will not have to make that distinction. I also want to point out that, look, we have a second process that is now starting right here. We have the development of a cleavage furrow, and therefore cytokinesis is starting. Now I want to make it clear, if I were to ask you to identify the structure here at E, I am looking for cleavage furrow because that's what this is. However, I could also point at E right here and say, what process is occurring here? And the answer I'm looking for is cytokinesis. Cytokinesis is a process. Cleavage furrow is a structure. Eventually, those chromosomes attach to the spindle fibers at the equator, a location also known as the metaphase plate, and we are now at metaphase. The centrioles then pull tension on the spindle fibers and pull the chromosomes apart, ana meaning apart. We are now at anaphase. The spindle fibers continue to reel in those chromosomes, and so we are still at anaphase. And as with prophase, you do not have to make the distinction between early and late anaphase. Anaphase is sufficient. Finally, the chromosomes reach their destination, and we have new nuclear envelopes beginning to form around them. We are at our final stage, or telophase. Also notice that our cleavage furrows are continuing to grow, so cytokinesis is continuing. Eventually, the cytoplasm is pulled completely apart, and we have now the formation of two new daughter cells. In ending, I just want to restate, study mitosis like the process that it is. Don't memorize. Literally sit down and draw the cells with the nucleus, if it's present, the chromosomes, the centriole. And as you're drawing them out, describe what is happening. You'll be amazed at how quickly you learn and master mitosis.